So good morning. Thank you very much for the invitation, Harriet and team. Thank you so much. It's a pleasure to be with you today talking about what can we do with the information that we have in electronic health records. So those are my disclosures. And now we are living in a time where we have a lot of information when we talk about cancer. We have all the omics and we have the capabilities of data processing that has been changing during the last years. So beyond that, we know that cancer is a complex disease, and we have a lot of different granularity of information that we can analyze. One of the main places where we have information from patients and diseases are the electronic health records. The truth is that from 2014, most of the different regions in Europe start changing their paperwork for electronic health records. So we have like libraries of how are patients doing in hospitals, and all those information, most of it is placed in free text. We know, of course, that AI is changing this situation. And AI is a big place where we have different areas, and one of those areas is called natural language processing and as well machine learning. And both of them can help us to extract all the information that we can find in these electronic health records in a way that can be automatically and not a manual chart review as it has been done during the last years. So why we do think that NLP took an important place now. And the truth is that you know that we have a structured information in the hospitals. We have, for example, ICD codes that may have information across different diseases. But the truth is that when we talk about cancer, for example, ICD codes are not going to give you the answer of how a patient is doing, how are the side effects, the outcomes. So we really need to look into the free text to understand what it is happening to each patient. And this is the information that it is going to be most relevant. There are some publications where they say that the 80% of the important information in cancer patients are in free text and not in ICD codes. Of course, it will depend on the region and country that we are working, because we know that there are several countries that they have a better informed structured data. But NLP is not that easy. And when we think as clinicians, I am a medical oncologist by background, when we think as clinicians are about clinical variables that we want to extract for the EHRs, the truth is that in our head, a clinical variable is a single issue. But this is not the truth, because a clinical variable is going to be built by different terms. And let me put you an example of that. For example, if we are thinking about anemia, we can look for the term anemia in a free text, or maybe we can go to a blood test and we can see that a patient has an hemoglobin of 6.5, and this is anemia. And we can also infer, for example, if we see that a patient have had a transfusion, that this patient has had an anemia. So it is really important from a clinical point of view to construct clinical variables based on different terms. And this is the way the information on electronic health records should be evaluated. We have a clinical variable or a concept. We need here clinicians that are going to do the curation of the different terms. We are going to define all the terms and inferences that can define a clinical variable, and this is the information that we are going to look for in the electronic health records. Once we have this information, we are going to build again the variable, and this clinical variable, it is going to be the one that is going to be placed in a clinical database. But it is not that easy as well, because you can say, okay, I want to look, for example, for a breast cancer patient. But the truth is that it is not like looking for breast cancer in an electronic health record. You need to recognize it. You need to, for example, understand that the patient is having the breast cancer is not a family background, so you are not seeing information in the family background. You need to understand that this is happening, and for that, you need to understand the context. And this is the most important part in natural language processing and the most complex one. For example, in our experience, we have had to develop a specific general models like the copy-paste. I am a medical oncologist, and when I was in the clinical practice, I used to copy-paste the clinical evolution of the patient and then change it for the new situation. This, for example, make crazy all the models because they don't really know if this is information that it is happening or not. So developing all these models, general models, to give context to what it is happening to the patient is such an important thing. And what are we doing? So finally, we are getting all this information. Of course, we can use as well structured information, and we are structuring all the information that it is going to be required to answer a primary objective for the study that we want to do. But here you have, for example, an example to show you that NLP is not that easy as well, and acronyms are a really big issue for NLP. You can see here, for example, multiple myeloma, millimeters, malignant melanoma. So how do we train the models to really understand that in that context is the real word that we want to analyze? The very important part here is that those models should be trained by physicians, are clinicians working at hospitals, so they can really train the different models how to continue. When we started doing that, one of the things that happens to us when we wanted to publish some data or some results of the studies that we were performing, it was that a lot of the reviewers asked us, okay, how do I know that the clinical database that you are analyzing is good enough? Because this is not based on the standards, that the standard is a manual chart review. So we create a methodology and we published it for other colleagues to use it, that we call it external clinical NLP validation. So every time that we are going to work with a specific hospital, what we ask is 3 people for that hospital work as annotators. Annotators are the ones that are going to check the free text to really see if the clinical variable that we want to extract is good or not. So we ask 2 of them to be annotators and 1 to be a chart, because sometimes there are a disagreement between the doctors as well. So, but we can then have a gold standard. So we compare what they extract with the clinical NLP extraction, and we have a metric to really guarantee that the clinical database is good enough. Of course, this is impossible to do with other clinical variables that we are going to include in a study, because there are going to be thousands of them. So what we do is that we define by protocol which are the variables that we are going to be externally evaluated. Most of them defining the population that we want to study, defining the outcomes, and also some confounders with the outcomes that we want to analyze to try to guarantee that you have at least an external validation. Of course, we do have internal validations as well, but we think that it is important to have external gold standard to be compared to. And for example, this is as well important because NLP is something pretty linguistic. So for example, it is not the same, the models that we are going to develop in English for UK compared to the US, because sometimes the language could be quite different and the models should work in both countries. And this is the way we approach to do a study with NLP. So we have a research questions, as we do have in any other study, a traditional study. We do need to create a protocol and a SAP. Once we have everything approved, we go to the hospitals and we ask them to participate. Of course, all of these protocols should be approved by an ethical committee to guarantee that the questions that we are going to do and the methodology that we are going to use are going to be approved. And once everything is in place, what we have created is a de-identification program. Because it is true that, for example, for structured data, it is quite easy to get out all the personal information. But we have understood that, for example, in free text, we can find personal information. It not should be like this, but it is. This is real-world data. So what, for example, we can see that we see in an evolution of a patient, if Mr. X get worse, please call her wife and the telephone number. This is something that we see in real. So we need some tools to really extract all the personal information. So we pseudonymized all the information in each hospital. They can use their tool or they can use ours, and all the personal information from free text is extracted. Once we have done that, we can integrate the data and then we can analyze it. Before analyzing it, we need to anonymize. This is something legal that, of course, maybe Elizabeth is going to talk about it later. But for saying from a legal perspective that this data is anonymized, we need to make another things. For example, one of the things that we do always is we change the day of birth of the patients. So we will have the month and the year that are going to be the real ones, but the day is going to be changed in order to guarantee that we have an anonymized database. And of course, there is no possibility to come back for the patient. So all the information that we are going to extract from this is going to be aggregated and anonymized, and it is the way where we can analyze this data. So we pass through all the general models and all the NLP techniques that I explained you before, and after that we get a big database that we analyzed, and then we can have our results to be evaluated and published. And here I want to show you some examples of what we have done since the moment in oncology. For example, this was a study. This study wanted to address two different questions for patients having cancer and having a thrombosis that were under an antithrombosis therapy. We wanted to create two predictive models, one to understand which patients are going to have another red thrombosis and which patients are at risk to have one bleeding. So you can see here in the slide how with this technology we could evaluate almost 3 million patients. For 3 million patients from the different hospitals that were participating in the study, we finally get a cohort of 21,000 patients that have cancer and have a thrombosis, and some of them were anticoagulated as a therapy. With this, we have created two different models. Here we have one of them, the one to assess the risk of having a red thrombosis. This technology gives us the capability to analyze different and a high number of clinical variables. For example, here we work with 95 predictors for the red thrombosis model. Finally, we get 10 predictors out to who were the ones who predict better, who were the patients that were going to have a red thrombosis. And most of the times here, we try to reduce the number of predictors as lowest as we can, because we know that for the clinical practice, it is easier to work with a lower number of predictors. And we can see here how now, for example, in the Spanish Society of Medical Oncology, these new methodologies of creating predictive models based on information from real-world data patients are starting to be mentioned. This is something that we are seeing as well, that clinical guidelines are changing, and they are also starting to recognize this work. Here you have another work in prostate cancer that was presented at ASCO. For example, in this analysis, we evaluate almost 20,000 patients with locally prostate cancer to really understand disease trajectories from the diagnosis to the development of the disease. And we did it with the same methodology that the studies before. Or here, for example, we presented at ESMO this year this preliminary data of a study that we are conducting in head and neck cancer patients. Here also we want to assess two predictive models. The first of them that was presented this year was the one for patients that were having a head and neck squamous cell carcinoma, and they were treated with a radical treatment to really assess the risk for their progression and really to try to understand which patients have a high risk to be metastatic or recurrence for trying to enhance their therapies. And here, for example, you have another one that has been presented in San Antonio this year for breast cancer patients with HER2 negative, and we wanted to assess side effects for those patients based on BRCA status. So those are some examples for you to see how NLP could really extract information and do it not manually, but automatically, so we can have these results. I want you also to explain that it is important to understand that, for example, in a research study like that, timelines could be different in some parts. All the regulatory part is going to be the same, and sometimes it is difficult. It is even more difficult because you need to have on board all the IT people. IT people is not used to work in research, but now things are changing. So they need to understand that they are part of this research, and this is something that could be sometimes quite difficult. So as now we have clinical units for clinical trial development. Maybe in some years we will have units that will involve IT people, and we will have these departments to really run with a specific process these AI-based clinical studies. And what is next? So I think it is important also because all of what I showed you before, it is based on retrospective analysis of patients to really understand what it is happening in real world. In real world. But the truth is that with all of that, we have an important tool that maybe it is going to help us as well to design clinical trials. And then we have here a really big opportunity because, first of all, extracting all this information could give us a real situation about disease natural history of the diseases and give you the real situation, so you have more information at the time that you want to design a clinical trial. Because most of the times, some clinical trials, when we do the statistical design, you based your standard of care on the clinical trial that approved that drug. But maybe the situation has changed now in the real world. So understanding what it is happening now, it is super important to design a clinical trial. Of course, for example, there is another important thing that it is synthetic or external control arms, something that it is also under evaluation by the different agencies, and of course, the possibility to include predictive models in the clinical trials to try to understand which are the patients who are going to have the better outcome, which are the patients who are going to have, for example, adverse events, so you can change your design. And here you have some examples of what we have been working. This was for ophthalmology, but the clinical trial Miracle, that was a phase 2 clinical trial, used our data from the real world in this disease to design the protocol to really understand what was the situation for this. We are also working a lot with different databases of the clinical trials. For example, if you have the data from phase 1 clinical trial or phase 2, and you also have the data of the population that you are going to, how the standard arm is performing in the real world, you can design and make some predictive analysis to repurpose this clinical trial design. And as I was explaining, also external control arms based on information of real world evident patients. It is an opportunity for rare diseases, or for example in cancer, for those cancers with low incidence or prevalence. For example, there are some works on glioblastoma, on uveal melanoma as well, and some cancers where the incidence is not big enough. And there is a new concept as well that is being discussed in the FDA and in the EMA, and it is called transportability. And it is something that we can do as well if we really understand what it is happening in the population, in the real world population. And for example, imagine a lung cancer clinical trial when you have, of course, your inclusion and exclusion criteria. But the truth is that sometimes you may apply this drug for the population that are not the same for the population of the clinical trials, because beside, for example, having the inclusion and exclusion criteria, the truth is that you may have co-founders, like, for example, smoking, that may be changing the drug effect at the end. So understanding, for example, how is your cohort of the clinical trials, how smoking is working as a co-founder in the outcome that you want to analyze, and understanding the rate of smoking patients in the real population can let you understand how it's going to be transport the effect of your drug to the real population after the phase 3 clinical approval. And this is my last slide. I just want to mention what it is most important for me here, and it is that AI and all these technologies are not going to work if we do not work together. There are all the technology part. When I started working in Savana, I learned a new language because I need to talk to the mathematic people and to all the physicians people that were working them, and they told me, Miren, this algorithm is perfect, and I say, okay, but that don't apply for my patients. So it is really important to have all involved, patients, technology, and all the medical and scientific part, because if not, AI will develop but will not make a difference for our patients. So thank you so much.